This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 1193, recorded on the 7th of February, 2025. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Montreal, Canada, Angela Mingarelli.
Hello, how are you today?
Is it really cold there?
Actually, it's warm. I know this sounds funny. -4°C is warm, but because it's been -20 for the last 3 weeks on and off, -4, I actually had my coat undone this morning. It was just open. I was like, it's not even cold. So everything is
-4°C here. Yeah, it's been like -12.
Mhm.
This is just balmy. Also joining us from Western Massachusetts, Alan Dove.
Good to be here. And it's 35 Fahrenheit 2C here, which is not too bad, except that it's blowing a gale out of the northwest. Wind is whipping through here. And we had some snow and sleet the other day, and now apparently Saturday night we're getting another snowstorm.
We don't have any wind here in the city. It's interesting.
We certainly have plenty here.
What was that wind thing that you picked once?
Oh,
Remember?
yeah, the global wind map.
That should show it, right?
Yeah.
We actually have quite a bit of wind in Montreal. I think it comes off the St. Lawrence, like from the river. It's very windy, so it always feels like 5 degrees colder than it actually is.
Well, if you enjoy these programs, you know, our main goal is to give you science, which is getting increasingly hard. You know, the MMWR just was published again after a little hiatus, and they censored some stories on H5N1. They pulled them out. To me, this is like stunning that they would censor science. But that's the world we are in. Anyway, we can try and interpret for you what's going on. Because there are other sources of information. And if you like that, please support us. Help us to do this. Microbe.tv/contribute. ASV, the American Society for Virology 2025 meeting will be held in Montreal at the Palais des Congrès de Montréal, July 14th through 17th, 2025. ASV.org/ASV2025. Check out the website for the meeting schedule. It's a bit different this year. Under the program tab, you'll see satellites on topics you might want to sign up for when you register, like emerging zoonoses, priority pathogens, plant virology, a trainee satellite workshop only for trainees on culture, communication, and mentorship, and a satellite on next generation virology, technology, and conceptual frontiers. And there will be a TWiV, too, on Thursday of the meeting.
Everyone is coming, right, Vincent? Or is anyone not making it?
I don't know who's coming, actually. We can't have everyone come because it's too crowded.
It'd be too crowded, yeah.
We have to have... I know that you will be there, Angela, because you live there.
It's true. It is on the way to my lab, literally.
If you would like me to come, I can easily get there. It's like a 3-hour drive straight up the highway.
That's true.
But I don't want to show up just to crowd the stage. So I think you'll probably have plenty of people.
Well, you know, these days not so many people show up, as you know, to Alan.
Yes, there is that.
So I don't know who's going to come, but I will send out a memo. How's that?
Yes.
I mean, Montreal, I know right now it's cold, but in July, it's beautiful weather.
Beautiful.
It's sunny. Montreal's a beautiful city in the summer. So if that can entice anyone to come.
Okay. A few news items.
Yeah. So I put a couple of these in. I can go ahead and probably
go ahead.
Take at least the first two. First item is there was a little accident at a major supplier of research monkeys in South Carolina, and apparently this is a facility that has had some other problems in recent years. They apparently had a piece of equipment somewhere, a heater maybe, that was producing carbon monoxide, killed 22 monkeys. And the USDA report on this cited it as a critical violation of protocols for animal care, but it turns out they're not actually going to do anything about it. I don't know why. So I've linked to an article in Science, but just USDA said, "Yep, this was a critical violation," and
Are these
apparently,
monkeys bred there? Do you know, or are they just?
I think these may be. I think macaques may breed in captivity. They
I
may
think it said
be.
it was a breeder facility for
Yeah,
scientific research. Like that's where then they send them to different parts of the country,
yeah. And this
which
is the
is
same
terrible.
place where just a little while ago they had a 43 monkeys escape.
Yeah, that's right. They
Which
Doesn't
they,
seem
is
they,
like the safest facility
pretty
either.
significant breach.
they got
That's
most of them, I think, right?
Yeah, they got him back, but this seems like maybe there are some more general problems there. I don't know
hmm
what's going on.
Hmm. All
Um,
right.
but that happened. Another item, the Animal and Plant Health Inspection Service of the USDA has been working with state animal veterinary departments, and they received data from Nevada on an avian influenza new genotype, D1.1 genotype. This is an H5N1 flu, but it's a genotype that had not previously been seen in cattle and has now shown up in cattle in at least Nevada. And probably if it's in Nevada, it's going to end up other places too. So bird flu is in the cattle, and I've linked to the APHIS site on that. And then in case APHIS goes offline, I've also linked to an NPR article on it. And then we have a couple of other stories that I guess Amy put in here.
Yeah. So we have a SIDRAP story. Argentina says it too plans to pull out of WHO by Stephanie Souchere.
hmm.
All right. Good riddance,
Yeah, I was gonna say.
Argentina.
I t-
This guy, this president, they screwed up the pandemic so much. Do we really need these supranational organizations that don't meet our needs? I think this is just unfortunate
when there is
because,
another pandemic, they will be very
yeah,
sorry because it's not even if. People are just like, oh, it's not gonna happen. It's not if. It's when there is. They will be very, very sad for pulling out.
one hopes that it is not in the next 4 years. Okay, let me
Hopefully,
just say that
Yeah.
also.
because it will be horribly mismanaged. You
Hmm.
will not get any information about anything except TWiV as far as we can wrestle it. So, I mean, the WHO is a good organization that helps a lot of countries that do not have the ability to help themselves. This is the altruistic model of humanity, folks. People help other people, and apparently the U.S. and Argentina now are backing out. I didn't sign up for that. That's for sure. I was raised altruistically, and I don't agree with people who think otherwise.
Hmm.
But a lot of people in this country are altruistic. It's just they got outvoted slightly.
Yeah.
Apparently, a lot of people sat out this election.
The problem, the problem is not the majority of Americans. The problem is that just enough of a population of people said, yeah, I'm not that into Kamala. I'm not going to bother to vote.
Yeah, now you
Um,
get what you
and
voted for,
now,
what you didn't vote for. Yeah,
that's
there you
so. If
go.
you're one of those people, thanks.
Another SIDRAP article. Lisa Schneering-Pejo, Pan American Health Organization, warns of rise of yellow fever in the Americas.
This is scary. They said there were 61 cases reported. 30 of them people died actually.
Yeah,
And
January. Yep.
yeah,
which
mostly
is
unvaccinated.
pretty typical fatality rates for yellow fever.
Most cases involve unvaccinated people.
hmm
So the answer here, folks, get vaccinated. But of course, these are countries where It may not be easy to get vaccinated.
And yeah, I mean, in this case, in vaccination, we've talked a little bit about the yellow fever vaccine on TWiV. This is not a flu shot. This is, it's a little more serious. I think people over 60 are not supposed to get it at all. Is that right?
Oh, really? I didn't
Yeah.
know that.
Yeah, like
Yes, that's correct.
there's
That's
an
right.
age cutoff on it
Yeah,
because
Severe side effects.
it's a highly effective vaccine, but it can have a pretty some bad outcomes if your immune system, depending on how it reacts, and apparently those odds go up as you get older, and so there are a lot of people who shouldn't be vaccinated, and those who are, it's kind of can be a rough ride. The real solution here is mosquito control, and that's a hard problem, but it is one that is addressable. Just needs to be done so consistently.
I mean, if they have access to vaccines, which I'm not saying everyone in South America does, but if they did at a young age, I would rather be sick for 2 or 3 weeks than
Yes.
die. So 50% chance fatality rate, I would rather have even if it's 2 weeks of,
Yeah.
flu-like symptoms, or even if it's severe flu-like symptoms, it's still better than dying.
Still better
if
than
they
dying.
can't. Exactly. If you're young enough and you have access, obviously, to the vaccine.
Yeah, but there are also a lot of mosquito-borne viruses in the same species of mosquitoes that we don't have vaccines for. So, a mosquito control program, which unfortunately is something that has to be diligently managed and funded year after year after year, and also ties into things like making sure people have adequate housing with screens, and the bugs can't just fly in all the time. All that together is also an effective way to manage it. It's why we have low rates of yellow fever in this country, because in weather when these mosquitoes are all over in the United States, but in the weather when they're out and about, we're charging from an air-conditioned car to an air-conditioned house, and doesn't get to us.
All right, we have some science for you today. 2 viruses we don't usually talk about. Alan, you're going to summarize that snippet for us?
Sure. So this first paper is Viral and Immune Dynamics of Genital Human Papillomavirus Infections in Young Women with High Temporal Resolution. So it's the viral and immune dynamics that they're going to get with high temporal resolution. The young women do not have high temporal resolution, as far as I know. And the authors are 2 co-first authors, Nicolas Tessandier and Baptiste Elie, and senior author is Samuel Alaison. And boy, are they from a few different places. There's the Université PSL in Paris, Université de Montpellier, Swiss Tropical and Public Health Institute in Basel, Institut de Génomique Fonctionnelle, Montpellier, University of Liège, Belgium, University of Manitoba, Winnipeg, Université de Franche-Comté in Besançon, which is where my grandfather was stationed in World War I. That's the only thing I knew about Besançon before reading this. Centre National de Référence Papillomavirus, which is in also Besançon. Paris-Saclay, Université Paris-Saclay in Orsay, CHU de Nîmes in Nîmes, France, the Sorbonne Université, University of Oxford, UK, I can pronounce that one, the German Cancer Research Center in Heidelberg, and the Public Health Agency of Canada. I think I got all of them. So this is a very international team. And to summarize this, I can actually just read the author summary on this paper. It's a PLOS paper, open access, and the author summary is actually pretty good. So they say, why was this study done? Despite human papillomaviruses, HPVs, being the most oncogenic viruses, we know little about non-persistent infections in young adults. Nearly 1 out of 5 women of age 25 has an HPV genital infection, but more than 90% of these clear in less than 2 years. The determinants of HPV genital infection clearance are largely unknown. In 2016, a longitudinal study was initiated to monitor variations in HPV virus loads and the associated immune response. What did the researchers do and find? HPV virus load exhibits a rapid increase, a long plateau, and a steep decrease. Infections are estimated to have a median duration between 13 and 20 months. Immune patterns are associated with HPV infection, especially gamma delta T cells and CXCL10 chemokine. And variations in virus load are primarily associated with HPV genotype and variations in infection duration with host differences. So what do these findings mean? First of all, HPV infection clearance is associated with the activation of the immune response. Okay, makes sense. Secondly, variations in virus load can help to optimize screening policies. Makes sense. And finally, details of the immune response can help identify biomarkers and targets for immunotherapies. So that's the basics.
Chronic infections by human papillomaviruses, they go on your lifetime, cause cervical cancers and most anogenital cancers and many oropharyngeal cancers. 83% of HPV-induced cancers are cervical, so women are most affected. There are 630,000 HPV-induced cancers worldwide, at least in 2012. A little behind on the statistics there, folks.
Yeah. 13 years.
And
Yes.
so these are sexually transmitted infections. But the good news is over 90% of these infections don't go on for more than 2 years. So they're acute infections. They're not chronic infections. And so why? Why not? And if you can find out why, maybe you can get some clues about why the ones go on forever, right?
And we should mention there is a highly effective vaccine now against the oncogenic HPV types that is now routine in vaccination programs in sensible countries around the world for boys and girls as they approach puberty, and that basically deals with this problem. The issue is there's a generation, actually there are a couple of generations of people for whom that vaccine came along too late, because after you've been exposed to this virus, after you've grown up and become sexually active, you've probably been exposed to this virus, and then the vaccine doesn't seem to be very effective at all.
All right, so that's what this paper attempts to do. A really unique study, I think, where they say, we have done randomized clinical trials for the vaccines, but they're limited because the follow-up was not all that frequent, and we didn't get samples that we wanted to, cervical smears in particular. So this study was designed to investigate the immune response, both innate and adaptive, to papillomaviruses in women, and then the viral load. They have a very specific definition. Viral load is the genome load, the number of copies measured by qPCR.
I
And
felt like they included that for us.
They normalize it to some human gene in the sample, which is the way you need to do it because every sample is going to be different quantity, right? And so this is called the PAP Clear cohort study. It was done in Montpellier, France, at 189 women, 18 to 25 years old, followed every 2 months, either until the HPV infection cleared or for 24 months, in which case they said, okay, it's enough, you can go now.
And this is, there are a few reasons why this was technically difficult, and we're going to talk about some of the techniques they used. This is not like a routine, simple thing to sample and figure out. So most of the HPV infections are going to be asymptomatic, which is why they had to do this cohort study and sample the same population repeatedly over time, because you don't know anybody's infected. The second problem, and I didn't realize this, but cervical smears, apparently you can't just freeze them and ship them off to the lab FedEx. They've got to be analyzed right away because it's a fragile sample. And the sample has a lot of keratinocytes, which are sticky and autofluorescent. And that's a big problem if you're going to do fluorescence-activated cell sorting, because usually what you do is you label your cells with some antibody against a surface marker on something and you send them through the cell sorter, and the ones that glow are the ones you want. But if you got all these other glowing cells in there, then you can't really do that. And that actually is something that they spend some time figuring out how to deal with.
Yeah.
But I saw that and I thought it was interesting that, like, I looked to see what flow cytometer they used, and that they weren't using, because there's spectral flow cytometers and then there's just like regular, like 4 laser, that don't use the actual spectral signature of the fluorophores, where every fluorophore itself has a very specific spectral signature. So if you use the cell type, like the keratinocytes, unstained as your just unstained control, it'll subtract all of that fluorescence from the cells. So they're talking about how they want to use CyTOF, but I thought it would be good, but maybe using spectral flow cytometry, and there's also spectral sorters, they might reduce some of that like fluorescent noise, because we do that. Like when I do liver versus lung in my control, I'll have an unstained liver versus an unstained lung because all of the cells have autofluorescence, and you use that to subtract from your samples so that when your actual fluorophores won't be adding to that. But it's still cool because I know that the material in a smear is very, very small as well, and they have very few immune cells. So the fact that they did this is actually very impressive.
Yeah.
Yeah, they had to use a lot of statistics because the numbers are small, and you could look at antibodies, and there's no clear. If you just look at the data, there's no clear trend, right? You have to put statistics through them to get a trend, and even at that, they say it's a little iffy, but it's what they could do. They had 974 visits by all these people.
That's a lot of visits, especially for a pap smear. That between all of those people, it is not the most comfortable thing to get done.
Yeah. Well, it's very interesting that last year I was in Stockholm and did a TWiV with a guy who works on HPVs, and Sweden has a goal to eliminate cervical cancer in the next 5 years. He said the only cervical cancer is caused by HPVs and nothing else. So they've eliminated pap smears in Sweden. It actually happened during the pandemic when people couldn't go. So they would send them swabs, and you would self-swab, and then they would just do a PCR. And he said, if it's negative, no worries. If it's positive, they would bring you in and look to see if there's a lesion. If there's a lesion, they treat it. If there's no lesion, there's nothing to do. So that simplifies it.
Does he mean? Sorry, Vincent. Sorry.
No, go ahead.
No, just that like there are other forms of, maybe not cervical, but I guess you can't detect like uterine cancers, different forms of uterine cancers. Those cells would never make it to the cervix, maybe. So he's saying exclusively cervical cancer. That's the only thing you can detect. Because pap smears, you can also detect infectious agents, right? Just other infections that you could potentially have. That's not just.
He said the only thing that causes cervical cancer is HPV, nothing else. And in fact, they even say that in this paper somewhere. And so he said we don't need to do pap smears anymore because if there's no HPV, there's no risk. If there is HPV, then you have to look and possibly ablate the lesion depending on the size. But if it's negative, and so he said in Sweden, no more pap smears. Can you imagine
that's kind of problematic, because like pap smear for HPV, yes, to prevent cervical cancer, yes, but pap smear in itself is not only for like determining if the person could have like mutated cells or cancerous cells or anything. It's just overall looking to see if there are other external lesions on the vagina. There could be other things, like it's a whole, because then people aren't going to be going to get like their, what do they call it now? Like just a checkup, which is for women every 2 years, includes a pap smear. And in that moment, they also check for other signs of like overall health. So there could be other things wrong. So if he's just saying no pap smears.
Well, they're presumably doing they're presumably doing the every 2 years get your exam. I would guess. So Sweden's healthcare system is certainly up to that.
They're very good. Yeah, I'm sure he's just referring to maybe that part of it they're saving
that
on.
particular test may not be.
Anyway, listen to the podcast. I was very impressed. I thought it made a lot of sense.
Yeah.
Cool.
All right. So here we.
But what are
back-
they? Sorry to continue the digression,
Yeah.
but it's virology still.
It's okay.
So
Okay.
what about women who are too old to have gotten the HPV vaccine? Are they, if they want to eliminate cervical cancer in 5 years, it seems like you're still going to have a lot of people around who could still get it, right?
Well, they do a PCR,
Okay.
right? And if you're positive, then they look in and see if there's a lesion. And if there's a lesion, then they ablate it, right? Laser ablation or something.
Okay.
Cryoablation. But yeah, they do treatment if there is a
Right. But okay. So
positivity.
that's not eliminating cervical cancer. It's in 5 years, you want to get to a point where there's no serious cases of it because you detected it early enough.
Well, between the vaccine and the
in younger
screening,
generations, you eliminate it.
Mhm.
Yeah.
right? If you vaccinate early enough, plus you do
Yeah.
the screening for everybody, then that
Yeah.
presumably
Okay.
does it.
That makes sense. Continuing then on the paper.
Yeah. So they all right. So they I'm gonna go through the methods, and then we're gonna go to the discussion for the conclusions, because the methods, the most of the results is basically saying how we did this, and the limitations and so forth, but not what it meant. So as we said, they want to characterize the local immune response. They're getting Pap smears, and they use flow cytometry. And as Alan said, keratinocytes are a problem. So they actually used software packages to put cells into clusters based on the fluorescence signal. And so, what does that sound like a reasonable approach, Angela?
Yeah, you can do that even there's a software called FlowJo that we all use to analyze flow cytometry data.
Yeah.
And you can put things into UMAP. Normally you do it when you have more than like 10 or 15 different markers. If you don't have that many markers, it doesn't really make to do the clustering. But yeah, people do this all the time. That's very reasonable.
Okay. So they identify clusters of cells in these samples, right? So initially, all they have is the immune data. They don't have the infection data yet, so they can't make any correlations. But they go through to tell you what we saw: mostly granulocytes in terms of white blood cells, and then we try and make an association between this cellular immune response and HPV status. And so we get people who had the same HPV genotype at doing 2 consecutive visits. So they end up with a data set of 145 HPV negative and 186 HPV focal flow cytometry samples. All right. They also quantify cytokines in these cervical samples using a package called Mesoscale Discovery. And we're going to get to what that means in a bit. And then, of course, the viral load kinetics. So there were 76 people for whom at least 3 samples had one HPV genotype during their follow-up. They had 164 infections, including 66 left-censored infections. It's so weird.
Yeah. So they use this term. I saw it in the
Weird.
abstract. I was like, many of the infections were censored. I was like, what?
Yeah, I know.
I also didn't know what they meant.
What they mean is, so some of these women came in and they already had an HPV infection at the first time point. So that is called a left-censored infection because you didn't see when it came up. You don't know how long that infection's been going on, and then, it resolves sometime later and you see the resolution of it. If the opposite happens, if they get infected during the study, but it's toward the end, and then the study ends and you don't see the ending of that, that is a right-censored. And then they have some that are double-censored, where they came in with it and they left with it, and just through the study they had the infection.
And then a complete infection, what would that be?
They came in without infection.
Got
the first
infected.
time point. Got it. You
then
saw
resolved
the
it.
virus come in, then you saw the virus go away
Okay.
before the end of the study.
What I found from this, it's interesting, is that 41 of the participants were co-infected by more than one genotype. That's very interesting. So I would not have guessed that, but people are multiply infected, so they multiply transmit, or you have multiple sexual partners. Actually, one of the requirements for this study was you had to have at least one sexual partner in the previous 12 months. And so maybe if you have more than one, you get more than one serotype, right?
Right, because if you did this in people who aren't sexually active, you're not going to see anything.
No, yeah, you
Um,
don't want to do that.
but
But that's where the multiple infections
yeah,
probably
and
come
the
from.
multiple infections could have a number of different sources. These viruses are they're durable. They kind of go everywhere. They're very contagious. It's one of the more contagious STIs, and since they're most of the time asymptomatic, I think it's not uncommon for them to kind of travel in little clumps.
Do we know if if people can get multiple infections from one sexual partner though? Has that been shown?
I don't know if
No.
I don't know if we have those data, but it's I had the reaction like Vincent, like, whoa, multiple infections. Then I thought, actually, that kind of makes sense from what we do know about HPV. You know, it's
Also, I'm sure sex workers are multiply infected, right?
well,
So
right,
they can transmit. I don't know how.
right, but I don't think you even need to invoke that. I think it's just
Maybe.
these viruses get around.
All right. So the virus data, they also have to apply statistics because if you look at the raw data, it's just like figure 4B. Oh my gosh.
Yeah.
Oh yeah,
Oh my gosh. It's like a kid took a crayon and just
scribbled. Yeah,
I don't know why. Yeah.
And then they have to apply statistics. And, they say we don't have a lot of prior data to give us some kind of model for this. So they had to do it themselves. So anyway, what they end up finding is very interesting. Infections last around 16 months. The plateau lasts about 14 months. So you get infected, you reach a plateau for 14 months. And then, interesting, there are a median of 2 virus genome copies per cell. And what they also find that's interesting is that most of the variance in infection duration, which is the scribble on that graph, is host differences.
It's interesting, this plateau that's that long, and they refer to it as like the immune system like waiting, but I think it's more just that there's not, I guess it's in a mucosal surface. We know that it's not systemic, so it's kind of like isolated there. Unless you have like actual lesions getting into your bloodstream, the immune cells aren't going to recruit as much, I guess. But I wonder how. Like, I don't know, like the immune mechanisms, I guess we don't really understand them, but that it can be there for that long. And I thought they've made it seem really interesting that gamma delta T cells were involved. Like it was like a new finding, but gamma delta T cells are involved in most mucosal surfaces, like in the gut too, and in like other mucosal infections. So it's like cool to validate that, but we kind of expected that in a mucosal surface, I think. Yeah.
So in terms of cytokines, they didn't see a strong, consistent increase or decrease in the cervical area. And there's a lot of individual variation among the sites. So it's very hard to get, any kind of correlations with cytokines.
Yeah.
and there's a lot of variation. We're looking at figure 4, but figure 3, where they've got graphs for 158 infections and 76 women. And if you look at this, it's not like, oh, here's the pattern. Exactly. Okay, this is kind of that, and this is kind of that, and this one has this. And so in all of these cases, it's a lot of statistical analysis to see if there are any consistent patterns and then to figure out what they might be.
And I think they only looked at co-infections for HPV, but not for other pathogens, because you could have so many, all of these cytokines. If I have, let's say, I don't know, some other like STI, sexually transmitted infection that is causing inflammation, then I'm going to have increased IL-1 beta or IL-1 alpha or whatever. And then somebody else won't, but those aren't necessarily related to HPV. And they're only looking at co-infections with HPV, not with every other thing that could be down there.
Yeah.
Which I thought was interesting.
Well, and every other thing that could be systemic.
Also,
right?
Also,
I mean, if you've got the flu, you're probably going to have some cytokines going all over the place, which would maybe be detectable in mucosa as well, even though the virus is not in a particular spot.
Hmm.
And they looked at viral loads and uh, local immunity, because they also look at some cytokines. So basically, they quantify circulating IgG and IgM against the 6 genotypes that they had been following in this study. And they detected no correlations between the viral load and seroconversion status, which compared. So there's a lot of people had no seroconversion, but if you did, it didn't really correlate with the viral load and the antibody titer. The one thing they can say is the mean concentration of this chemokine CXCL10 was negatively correlated with viral load. That's the one thing that they come up with here. This gets a strong signal. So let me summarize this for you. These HPV infections, now everything they conclude is based on models, okay? HPV infections have 3 phases: a short growth phase, a plateau, a long plateau, and a short clearance phase. And so that's the observation, which is very interesting. Why that is, we don't know. The immunity studies don't really answer that, but give you some suggestions. This dynamics is contributed to by the genotype and the host. Both the genotype and the host contribute to that dynamic. Differences in the dynamics, for example. They find a role for granulocytes in these patterns, which they say others have been found, and gamma delta T cells, as Angela said. They have 3 distinct subpopulations with those cells. They have a lower proportion of CD4 T cells in individuals with focal HPV infection than an uninfected individual. So they say it probably doesn't have any role. Okay, no role in controlling infection locally. And then the dynamics of local immunity. And again, they have clusters of innate immune responses. And I said already, granulocytes increased in frequency early in infection. And they say this supports the idea that the immune system can detect HPV early on and react to it. But they say clusters associated with the adaptive response increase in frequency later. So
And I
there's
guess
some
because
delay.
there's not really antigen, I guess, there, like eliciting recruitment of immune cells or adaptive immune cells. But
Yeah.
the whole granulocyte thing I thought was interesting. I guess they must be limited. They were only using CD16. They weren't like differentiating what types of granulocytes, like neutrophils, eosinophils, mast cells, basophils, which all hang out there anyway because they're protecting the mucosa. So I wonder, and it didn't really look that significant, granulocytes, between like uninfected versus infected people with HPV, unless I'm misinterpreting the data, but it seemed like there were already lots of granulocytes there and there was a slight increase, which is surprising that it's like, that it's not more.
All right, so then they say, okay, can we take these data and interpret how infection could unfold? And they say, okay, this striking pattern is that CXCL10 is associated with milder infections. And
CXCL10 correlates with the innate immune response.
Yeah, the early. And then they say we find immune cells associated with adaptive responses peak later in infection.
Which, like, makes sense.
Yeah.
So they say one idea here is that a fraction of HPV infections evade the innate response, like interferons, and they establish a persistent infection. And then the adaptive response kicks in later, and maybe that helps to clear it, right? And these clusters of cells that they're observing, maybe they're involved. So you can't really do any experiments, obviously. What we could do is statistical predictions, right? And I don't even know how you would test any of this, frankly.
I mean, I think the coolest thing from this paper is showing this like plateau phase that we didn't really know about and the clearance
Yeah.
at 2 years. Like, I mean, I didn't know that. Maybe this was already known, but I think that's pretty novel. The 16-month average
Yeah,
plateau
it's
phase. That's
interesting,
interesting.
Yeah, it definitely is. And I think that's not something that people had looked at, certainly not in this level of detail, because again, these are asymptomatic infections. Nobody knew they had anything. Nobody is checking for this other than through pap smears, which are only going to show you if it's
Yeah,
symptomatic.
Hmm.
So I think that is a novel finding here.
Yeah, that's cool.
And yeah, I mean, it's going to end up being a resolved infection, but it sticks around for a year and a half. That's kind of weird.
It is.
Question is, can they actively transmit during that time? I'm guessing yes. Do we
Probably.
know?
There's
Maybe?
there are
Yeah,
virus
I
genomes.
guess
But
so.
depends
Yeah.
like how much. It'd be interesting to know if they were potentially like if they could transmit to another
Well,
Right.
I mean,
individual.
it has to be that these are the people transmitting, right? The ones who are infected for whatever months.
But I
Has
mean
to
like
be.
closer to the end, like at what time
That's a good
would
question.
they be considered like cleared versus not? Do they
Yeah, I
Right.
don't know.
do they have those data like closer to the end, like as of 16 months on average or 18 months? I don't know.
Well, given the crayon scribble variation in viral loads, I'm sure there's going to be a distribution of people who are in that plateau, but they're not really contagious because their virus levels are low, and others who are in that same plateau, they've got sky high virus levels, and maybe they're more contagious. But given how hard it was to even figure this much out, I don't know. You're going to need a really big, long, invasive study to figure
Sure.
that out, I think.
Even this is impressive. That many
This
people
is
every
very,
2
very
months.
impressive.
It's
This
very
is why
impressive.
It's a
Very
cool study.
impressive.
So the other thing they did, they knew who was vaccinated here,
Yes.
and they did not see any infections of vaccinated people with the same with the genotypes that are present in the vaccine. And so, but they say any effect of vaccination on, which they did see on infection duration or plateau viral load, is probably from cross immunity, right? Which they did see in vaccinated people.
Good to know. Get vaccinated if you can.
Yes.
So yeah, their analysis of kinetics showed no association with vaccination status. However, they observed the trend for participants being already seropositive for a genotype before infection by this genotype, whether by vaccination or prior infection, to have smaller viral load than those who never seroconverted. So if you're already seropositive by this genotype for which you're positive by vaccination or infection, it's a smaller viral load. I don't know what that means, frankly. And then finally, weak evidence for the mean IgG from vaccinated individuals to be negatively correlated with viral load, again, because of cross-reactivity.
I did like how when they're talking about this in the analysis, they did not detect any, they put it in quotes, breakthrough infection, meaning a focal infection of a vaccinated participant by one of the vaccine genotypes. I appreciated the scare quotes around.
They must listen to TWiV. I
They
feel
must.
like
I'm
these are
pretty sure these are TWiV listeners on here.
Well, they know that it would be interesting to study these kinds of infection in the same way that they've studied here, right? To see people, that would be really hard. You'd have to have a huge cohort, I think. But you say people who have received these genotype vaccines, and do you see any infections? And if so, what's the immune signature of that as well? But they said this make this statement: natural immunity may protect against HPV infection, but not enough to prevent it entirely. I find that a weird sentence, right? May protect against infection, but not entirely. So this is where people get into trouble, where they say protect against infection, but they don't mean entirely protected.
Right.
They just mean partially protected. So,
But also, what does that mean? You still get infected, so
Yeah, you get
potentially.
infected, yeah.
So
Yeah.
what?
Maybe you
takes
get a shorter
longer
course
to,
of infection. You get less
yeah.
virus production. Yeah.
Hmm.
I mean, I'm interested because I'd like to know, in vaccinated people, do you ever see infection? They notice in this study, which is very small, they don't see the same genotype,
Right.
but they do see effects on other genotypes. So I wonder if you did enough if you had enough people in the cohort, would you see infections of the same genotype as you were vaccinated against? I don't see why not, right? It's just, it would not, maybe the infection would be very brief, right?
Mm-hmm.
Because you have antibodies or whatever kind of immunity.
Mm-hmm.
Anyway, I thought this was really interesting because I don't really think much about, you think about people get HPV and it's cleared, and then you have HPV chronic and you get cancers. Well, what about in between, which is what this is, this acute infection? And there's some hints here about what's controlling it, but there's still a lot of work to do. Meantime, get vaccinated.
If you can, if you're not too old.
Mm-hmm.
Yeah.
Yeah.
I mean, you could get HPV. It's not like yellow fever vaccine where you're worried about, some bad outcome if you're too old and get vaccinated. It's just it's too late. Yeah.
It's still,
For a lot of
yeah.
us.
It's really unfortunate. There's a big attack on HPV vaccines in this country. There are lots of lawsuits um, coordinated by you know who.
Yep.
There's a lot of attack on a lot of vaccines in your country, Vincent.
Yeah.
So I feel
Well,
like
HPV in
is any
particular
vaccine safe?
HPV in particular, there are a lot. Even though many studies have shown that there's no autoimmunity, there's no this or that. But if you can get into a civil court with, all you need to do is convince the jury. You don't even have to convince them of the science. You just say, look at this person. They got this after getting vaccinated, and that's it. The jury is likely to make an award. And so the problem is right now, there's a vaccine compensation fund where if you claim you've been injured, they review it, and then for HPV, they don't give awards because there's no
There's no
association.
documented harm.
And there's no harm. Yet when it gets to civil court, and the head of HHS can make a difference in how often these go to. It could be that, like what happened in the 80s, many vaccine manufacturers may drop out because they can't afford this, the costs of this litigation, right? In the 80s, like, the manufacturers of vaccines dropped precipitously until Reagan signed into law the Vaccine Injury Protection Act, and so that could be gone, and so women who benefit incredibly from HPV vaccines may have trouble finding them. So I'm very upset about this.
Yeah.
Just the whole politicization of science and vaccinology in particular is unacceptable. It's unacceptable. And, we got letters from people who said, you better be political because that's the only way that you're going to be heard. Okay, another cool paper here. PNAS. Summary, please.
Yeah. So let me get to my summary and my tabs open here.
Another
Right.
virus that
So
if you get it with age, it can also be a little bit more severe.
Right.
I was just adding that.
Right. So Epstein-Barr virus, BALF0-1, BALF0-1, subverts the caveolin and ARAD pathways to target B-cell receptor complexes for degradation is the title. This paper is not open access, unfortunately. The co-first authors are Stephanie Pei-Tung Yu and Yifei Liao. Senior author is Benjamin Givers. The team is from Brigham and Women's Hospital in Boston. So I should say this is an all Cambridge-centered team. So Brigham and Women's is in Boston, Harvard Graduate Program of Virology in Boston, Broad Institute of Harvard and MIT in Cambridge, Harvard Medical School in Boston, and the University of Cambridge in the other Cambridge, UK. So as a quick summary, somewhere north of 95% of humans carry Epstein-Barr virus, which can cause mononucleosis. It can trigger multiple sclerosis. It can also cause various cancers in some people. It is a herpes virus, and like other herpes viruses, it has two possible modes of infection in a cell, lytic and latent. During the latent phase, EBV hides out right in the immune system in B cells, which is pretty gutsy. This paper focuses on the lytic cycle, where EBV reactivates from latency and starts actively infecting and killing cells again. In order to do that, it has to somehow prevent the B cells in which it lives from mounting an immune response that would stop it. Using a bunch of cool proteomic and biochemical techniques, these folks found that an EBV-produced protein called BALF0-1, or BALF01, I don't know what we're going to call it, targets
Just BALF.
BALF.
Maybe
Yes.
easier.
It sounds like we're throwing up, but BALF targets B cell receptor complexes and directs them to the cell's proteasome for degradation. Besides revealing potential targets to intervene and stop EBV reactivation, this also suggests a possible new strategy for chemotherapies against lymphomas that depend on B cell receptor signaling. So that's my short version.
There is a EBV protein called BARF.
BARF.
Yeah, I was going to say it was BALF and BARF because they also mentioned it in the paper.
Yeah.
Right.
Very funny. Very funny. Yeah, so this is cool because you get infected with EBV, as they say, 95% of adults are infected early in life, and the virus infects epithelial cells and then infects B cells and makes them into memory B cells. So they're just quiescent, sitting there with antibody on their surface, and the genome is in there, and a couple of viral proteins are made.
It's a little like a bank robber hiding out at the police station.
It's very interesting. And these
Kinda
B cells
scary.
can hang out anywhere. They could be in the bone marrow. And then periodically they reactivate, which means they start making virus. There's this, as you'll see in the paper, there's this idea that somehow there's cross-linking of these B cells, memory B cells have antibody on the surface, right? Which, if it recognizes its cognate antigen, will activate the B cell so that it starts cranking out antibody. But if the antibodies are cross-linked, it will turn on EBV lytic cycle, will make virus, and then that virus will go back to your epithelial surfaces and reproduce out and spread to somebody else. So it's a very cool strategy where B cells are essential. In herpes simplex, neurons are essential for latency, but for EBV, it's B cells. And the cool thing is that it makes them into memory B cells. So as Alan said, how does a virus avoid this antibody on the surface?
Right. So some of the B cells it infects are going to be potentially B cells that are responding to EBV.
Yeah. It turns out there are a lot of your B cells after an EBV infection are EBV antibody positive. Yeah. So that's a problem, right?
Yeah. So how are you going to get out of that?
It's always a way.
Yes.
Which is
This
what we're
paper
gonna
was
find.
very cool, but some of the technique, it was dense to get through all their experiments, and there's so
Yes.
much work in this paper. It was very impressive,
Yeah,
but
it's
the
very,
amount
very
of
deep.
experiments they did and Western blots, wow.
Yeah,
I
it's
would
a
never
lot.
want to have to do that many Westerns, but it's really cool.
All right. So how would the virus? So the idea is that, or so they call this the B cell receptor. The B cell has antibodies on its surface. You can also call those B cell receptors, right? But they're basically antibody molecules stuck in the membrane, and they're specific for one epitope. And if the cell finds that epitope wherever, it's gonna get activated. So they looked at interactions among viral and cell proteins, and they find interactions between the heavy chain of immunoglobulins and two Epstein-Barr virus early lytic proteins, BALF and BARF. BALF01. Well, we'll just call it BALF and BARF. And also with multiple proteasome components and proteins involved in transport. So this is intriguing, and they want to know what's going on here. So said the first question they ask is, these two proteins on their own, could they get rid of the B cell receptor on the surface of B cells? So they produce the protein in cells and on their own, and only BALF. So the on the surface of these B cells, not only is there antibody, the B cell receptor, but there's also CD79 and another CD, which
CD79A and CD79B, I think it
B.
was. Mhm.
So when they put BALF in these cells, it depletes 79B and the antibody heavy chain. So that's interesting that the protein can do that. So they ask whether both can each target for degradation, and either one, expression of either one is actually enough to deplete IgM and CD79A. Okay, so what is degrading this? There's some interesting drugs in this paper. Bortezomib. Bortezomib. Who the heck would name that? So it's a proteasome
Same people
inhibitor.
who came up with Comirnaty.
That's true.
Comirnaty. Cells have
That's true.
a garbage disposal called the proteasome. Big protease in the cytosol that, well, it degrades proteins that are targeted for degradation by a modification called ubiquitination, but they also will chop up proteins for loading onto MHC and presentation and so forth. So kind of important. Proteasome, they have an inhibitor, and when they add this inhibitor to cells that make BALF, you get more antibody on the surface. So this is inhibiting the removal of antibody from the surface. And this is a paragraph with 14 different experiments in it. Oh my gosh.
It's one paragraph in the paper.
It was hard to keep track. I had to read so slowly.
Then they have CRISPR experiments, right, where they knock out BALF, and they show that is important for downregulating the antibody from the surface. And so they conclude from all of this. I'm not going to go through it all. It's too much. I want to give you the bottom line here, that BALF somehow associates with these B cell receptors and destabilizes them. How it does that, we're going to find out. How it does that, we're going to find out. So somehow the BALF has got to take the antibody from the plasma membrane and get it inside the cell where it can be chopped up, right? So they're thinking, well, maybe some kind of endocytosis. And there are lots of kinds of endocytosis. They use microscopy and fluorescence to study which one. And it looks like endocytosis is involved in general, but it's not clathrin mediated. So endocytosis is defined based on protein markers. And one of them is clathrin, and the other one is caveolar. Angelo, I always think of pasta when I think of cavioli.
So did I. I thought the exact same thing, and I was like, wow, I'm so Italian. And I didn't think anybody else would have thought of that.
Cavatelli and yes.
So the marker for
Cavatelli endocytosis. Yeah, I like to do that.
The marker for caveolar endocytosis is caveolin, which has got to be a pasta, right?
It sounds like it.
Anyway, there's also inhibitors here. So inhibitors of clathrin-mediated endocytosis do not block the removal of antibody from the cell surface. But they have an inhibitor. And that inhibitor, by the way, is chlorpromazine. Sounds familiar. There's a caveoli inhibitor, genistein, that inhibits the downregulation of the antibody.
Because calveoli is calveolo. How do we say it now? Oh my gosh. Caveoli basically is just like a shuttling system. No, it'll take it like from the plasma membrane. So
caveoli endocytosis is interesting because it brings it to the ER. Whereas clathrin mediated just brings it into the cytoplasm, right? And they fuse with lysosomes. But interestingly, caveolin targets the ER. So that's going to become important here as we will find out. They also deleted caveolin the main structural protein of caveoli. And again, if you delete that by CRISPR, you inhibit the ability of BALF to get rid of antibodies from the cell surface. So the conclusion is BALF triggers B cell receptor internalization via the caveolin pathway.
Right.
Which that in itself is so cool. Also slightly horrifying, but very cool that the virus is
Yeah, it's great.
internalizing the BCR like to hide itself from the immune
Yes.
system.
Yeah, it's amazing.
Take in the
Crazy.
sign quick so nobody
Literally.
sees.
Yeah, bring down the flagpole. Bring
Yeah.
down the flag. Like,
So then, this confused me for a long time, but I think I figured it out. So if you cross-link the antibodies on a latently infected EBV cell, it will activate the lytic cycle. And they want to know, is it the cross-linking that is downregulating the antibody, or is it the actual lytic cycle of EBV itself? And it turns out it's the lytic cycle that you need. It's not just the cross-linking, but induction of the EBV lytic cycle induces internalization of these B cell receptors into caveosomes. It's cool, right?
Yeah.
All right. So how does this get the antibody degraded? Well, what they're thinking is these antibodies, these B cell receptors, are ending up in the endoplasmic reticulum because this is the caveolin pathway, and there is a known pathway where misfolded proteins are taken from the ER to the proteasome. It's called the ER-associated degradation pathway. I think that's in the title, ERAD,
Yeah.
of the paper. So basically, if proteins are misfolded in the ER, which happens a lot, this pathway will take them to the proteasome where they're going to be degraded because they're misfolded. You don't want them.
Sends them to the shredder. Yep.
it's like a quality control. So now
Yeah, it's quality
the virus
control,
is like, oh, let's throw out these BCRs. We don't need these.
Yes. So basically, that's what we're going to find out is that
Sorry.
the virus
Spoiler.
it says it slips these BCRs into this ERAD pathway.
That's so cool.
And they
Yes.
get degraded. So they do a whole bunch of experiments to prove this. There's this protein that's important for ERAD function, and if they knock it out, it reduces the degradation of the antibody. They have an inhibitor of ERAD called EOR. It's called EOR. EEYA. Earistatin. Wow.
Yeah, it's a
That blocks the pathway, and that also impairs degradation of the B cell receptor. And gosh, every paragraph is like 20 experiments.
Yes.
It's just remarkable.
it
It's just
is.
remarkable. I mean, there are also controls, which I'm not mentioning, which
Right.
are really important. But
It's true.
the
The
key here
results
is-
started with, I think, at least like a whole page of just the supplemental to get to figure one. I was like, did I miss
Yeah.
something?
Yeah.
I was reading and I was like, where are they mentioning figure one? So much work was put into this.
all right. So now the ERAD pathway is involved. So now they have to say, prove that the proteasome is involved. Okay. So the way that things get targeted to the proteasome is they have to have ubiquitin. One way is to have ubiquitin, a little molecule stuck onto them, covalently linked, and that's like a marker to go to the proteasome. And so they find that they want to know whether BALF is enough to induce ubiquitination of these B cell receptors, right? And the answer is yes. You can see they get ubiquitinated. They have an inhibitor of ubiquitination. The first enzyme in the pathway is called ubiquitin activating enzyme E1 ligase. They can inhibit that, and that impairs BCR degradation. And so all these experiments prove that BALF uses the ERAD pathway to get the B cell receptor ubiquitinated. Remember, it's now taken off the cell surface. It's in the ER. It's going to get ubiquitinated and boom, going to proteasomes. So that's the mechanism. But how does this happen? Like, how does BALF recognize B cell receptor on the cell surface. All right, so they say, let's look at the cytoplasmic domains. So CD79A and B have long domains, 61 and 41 amino acids. And immunoglobulins have 3. And they say, it's got to be the longer one. Nope,
Nope.
it's not. They can deplete CD79 or 79B, and it has no effect whatsoever on the downregulation of the B cell receptor, right? So it is downregulated along with antibody, but it's not important. It's a bystander, because if you get rid of it, because they're thinking the BALF is sticking to the cytoplasmic tail of these proteins, right? But no, it's not. You don't need these proteins at all. So much for that. So then they turn to the antibody, which has got a 3 amino acid cytoplasmic domain. They go, well, this must be important. So they take out these 3 amino acids, and guess what? The antibody stays on the surface. And they think they're 2 lysines, and lysines are typically ubiquitinated. So they think maybe these are being ubiquitinated eventually. But that's BALF is binding to this 3 amino acid cytoplasmic domain. It's very interesting. Okay, now this is where it gets even cooler. So when Epstein-Barr viruses mature in the cell, so the DNA reproduces in the nucleus, and then it acquires membranes and traverses the secretory compartments on the way to the plasma membrane. This is the same pathway that antibodies use to get to the cell surface. Right. So they're wondering, could that be a problem, having antibody going in the same vesicle? Say, if it's an antiviral antibody, that would be not good, right? And so maybe BALF's function is actually to also interfere at that step, right? To make sure that the antibody is not binding to virus. And so they do an amazing number of experiments using a monoclonal antibody against an Epstein-Barr virus protein, which will neutralize virus infectivity. And if you don't have a BALF around, you get much less infectious virus produced compared to if you have BALF around. So this BALF is doing a couple of things. Is getting antibody off the surface and degrading it, but that is probably so that it doesn't interfere with virus production. And they actually showed that this BALF prevents the incorporation of the antibody into the virion envelopes. Otherwise, you would be making virus particles with antibodies stuck on, destined to fail.
That's not going to work well. No.
Yeah, already doomed, right? Because you have antibody. I mean, the cell is trying hard.
It's doing its best.
Yeah.
But there's always a countermeasure from virus, and this BALF1 this BALF protein is really important for countering that. And there's a very nice figure that there's one more really cool experiment, but there's a very nice figure showing you in B cells with BALF1, the antibody on the surface is taken in and ubiquitinated and brought to the proteasome and degraded. Now, in those same cells, EBV is being produced, right? In the absence of BALF1, they would have antibodies stuck onto them, and they would be the antibody would go on the surface, and the virus would be stuck to it, and it would never go anywhere. It's really nice. All right, so it turns out that certain cancers, certain non-Hodgkin lymphomas, need BCR signaling to survive. So the BCR is on the surface of the B cell. These are B cell cancers. They need signaling to survive. So they're thinking, hey, if we put BALF in, we're going to get rid of the BCR. There's not going to be any signaling. What's going to happen? So they did it, and the tumor cells die.
Ask your doctor about BALF.
This is so
Well,
cool,
this
right?
is all also this is all in vitro. Nothing has
Yes,
been
yeah.
done. So we don't know
Yeah.
what BALF would do in an organism, in a mouse, even
Right.
in a human, let alone a human.
No,
But
I'm pretty sure you don't want this in all your B
yeah,
cells.
still very
But
cool that it happened in vitro. Very cool. But what does that mean and how can we use
Right.
they
that?
say we need a model. We need an
Yeah.
animal model to test this. But it's cool. You put BALF in these cells, and
Hmm.
it triggers cell death because there's no more B cell receptor dependent signaling. The B cell receptor is gone. Isn't that cool?
Yeah.
And I don't
Love it.
think they said anything about like did they measure like interferons or anything in the cells? I don't think so, eh? Like to
No, they
see
didn't. They didn't at all. Yeah.
because I wonder if BALF in itself is immunogenic. It must be. Like what type of immune response, if any, would it elicit?
Yeah, it's a good
I don't
point.
know.
They don't talk about that at all.
Hmm.
So,
TBD when they do it in mice.
this is cool. They say as a B cell tropic herpes virus, EBV must cope with large quantities of immunoglobulins. And they say, here it is. It's BALF. That's how it copes with this protein called BALF. It facilitates release of cells that express antibody. So again, these B cells are making EBV, right? The lytic cycle is turned on. They're making EBV. You can't have this antibody in these cells because it's not just on the surface. It's inside the cell making its way up, and it's going to stick to virus. So this is a brilliant. That's why the BALF is there, so that these antibodies don't neutralize virus infectivity. Because if you're gonna replicate in a B cell, you need to have the right equipment to do that. Now, they say here, these findings raise the question of how frequently does EBV infect a B cell that has an antibody to one of its proteins, right? You may think, that can't be very common. It turns out there are germline encoded EBV antibodies, and they're more frequent than you might think. And in fact, well, at least for T cells, 20% of tonsillar T cells can be reactive with EBV encoded peptides. So they're thinking that there are a lot of B cells that could react with EBV. So this protein has emerged to target just this one aspect, which is really important of EBV production. I'm just thinking, what if you could make a vaccine lacking this thing? Would that be a good vaccine? I don't know. BALF 0. Remember, we've done this paper on TWiV. EBV is a risk factor for MS, right? It's a trigger for MS, quite clear. And so we need to prevent people from being infected somehow. I don't know how you do that. But anyway, cool, right? Isn't that a cool
Very
paper?
cool.
I like that very much. A biphasic life cycle. Now, there's a lot of molecular biology here, which is unusual for us to do these days. We used to do it pre-COVID. We used to do a lot of these papers, but I think it's very cool. It tells you basic, fundamental molecular biology of how viruses reproduce and evade. There you go. EBV.
Really cool.
Let's do some email. Angela, can you take the first one?
Yes, 1 second. From Joseph. Joseph writes, "Hello, TWiV team. I would like to express my sincere gratitude and sense of honor to have our recent paper on rotavirus, NSP4, and intracellular calcium waves reviewed on the TWiV podcast this week. It was an excellent summary of the paper, particularly delineating the intracellular versus intercellular calcium signals. You all asked some really excellent questions, and I will work this weekend to write up succinct answers to them both for your own edification and in case you get questions from TWiV listeners. As a teaser, I am giving you the plenary lectures on July 15th at ASV 2025. Some of your questions will be answered in that talk. Stay tuned. Thank you all again for your contribution to virology, education, and communication, and thank you for highlighting our work. It's a huge compliment. Best, Joe." And Joe is a PhD associate professor at Baylor College of Medicine. Baylor, sorry.
It was a very nice paper.
Yeah. Yeah, that
Yeah.
was a neat one.
Calcium waves. Alan.
Catch the wave. All right. Fernando writes, Hi, TWiV Inquirers. At the end of 1189, awesome episode with the calcium waves, you speculated about how the deep snow road trenches in Japan come about. As a twice delighted Hokkaido deep powder skiing visitor, let me enlighten you with a YouTube video. And he sends a link, and I watched this. And the answer is it's a really, really big snowblower, and it's really cool. Says that's how it's done. Our own California Sierra Road and rail snowplows are not far behind, and sends YouTubes of those. Finally, I wonder if the choice of Hokusai's Great Wave off Kanagawa as the episode image was triggered by the Wave Plus Japan Association. Was it? Not consciously?
So I was looking for something of a wave, right? And I searched for wave, and then lots of beautiful ocean waves. And then I saw that, which is a gorgeous piece, right? And I said, I have to do this. But I must have been subliminal because of the Japan thing with the snow, right?
Yeah, it's a famous Edo period block print.
Love it. Just love it. Yeah.
Fernando continues, I love digressions in the midst of serious virology. Always learning. Longtime supporter, Fernando. Thank you.
In fact, there's a very good video. Who is the famous, not Hokusai, but there's another artist of the same era. Anyway, he took a trip from Edo to Kyoto, I think. So Edo at the time was the capital of Japan.
Right.
Right. And he took a trip, and this was a trip that the samurai used to take all the time. So he went with them along on this trip, and he stopped at all the little places and made images, right? And some very famous images came out of that. And they look very much like this. Same style, right?
Yeah, same.
I love it. So, I love this style, and I think it probably, the wave, and it was clearly the best wave thing that showed up. And it must have been the snow also that triggered it. Yeah, I like that. Arjan writes, same thing. You probably received more responses to how the Japanese snow walls are made, but in case, here's a short but clear description. This one is a website. The snow walls, how the snow walls are made. The snow walls of Tateyama Kurobe. And lots of maps and pictures. And there's the big, lots of heavy machinery.
Yes.
Look at those things. The road must be located under the snow. They're using GPS to map its exact location. A bulldozer is used to scrape off the top layers, and then these snow blowers start spitting out. What a science, eh? I mean, I guess they want to have this road open.
Yep. Yeah.
And the highest it's ever been is 23 meters.
Wow.
That's pretty high. I love it. Thank you, listeners. I love when you get involved in our stuff, especially our off-road stuff. Okay, back to you, Angela.
Yes, John writes, interesting about why we don't vaccinate chickens. Given the number of chickens culled, is keeping some apparently significant % of the export market still worth it not to vaccinate? Numbers for how many birds have been culled are all over the place, but in trying to find out, I learned that the USDA compensates farmers for birds and eggs lost. There are probably a lot of underlying details there, but maybe that's why there's little pressure against not vaccinating. But I'm going to guess that the chickens in the stores now are mostly from vaccinated chickens, and that the future supply of chicks will come from that source too. So here's an idea. Label egg cartons with, in all caps, these eggs laid by chickens vaccinated against H5N1 bird flu. That gets a further message out there for vaccination, and it will also decrease demand a bit from the anti-vaxxers that won't buy them, helping to ease the upward pressure on price. Best regards, John.
So I think, wasn't the reason they don't vaccinate because it's a problem with export?
Well, yeah, exactly. Trades, and also they can be asymptomatic infections. There's so many things that you can't control with animals. Like when stamping out in like culling of all the animals is very necessary, because you don't know if another bird also that's flying in, migratory bird, is then taking that virus to then another, down the road to another chicken farm. And it's just, people don't realize how many secretions animals make and how easily like they can transmit from one another. Like if you see a chicken cage, there's like 20 chickens in a cage. Right next to it, there's another 20 chickens. Right next to it, there's another 20 chickens. If one of those chickens are barely even going to cough, you won't even know that they're sick until they're all infected with influenza. And then some of them might already be going out to market or wherever. And then there's contaminated meat. And then you're sending like possibly contaminated meat to another country, exporting it. And well, globally, it's illegal. Like no other countries will accept the meat if the chickens aren't, like if they were identified with bird flu and then vaccinated against bird flu. It makes no sense. They're just all killed, unfortunately. And we need to just control the spread. Keep your chickens inside. Don't let them get near geese, migratory birds, water.
So you guys do you have chicken culling in Canada?
Of course, yes. It's something that's done worldwide.
Yeah.
And it's not only chickens, it's cows, it's pigs, it's any sort of infectious disease that is, if you do something called like stamping out or like a cull, every single animal has to be killed. And like with the African swine flu, for example, in Europe, it's not even just your farm. It's every farm within, I think it's like a 30-kilometer radius or something. It's that there's different depending on what country you live in. There's X amount of kilometers from where that infected point was. Like even with foot and mouth disease, it was like that for a long time, that the radius around your infected farm also had to kill all of their animals to assure that it hadn't spread.
There's also, so that's an excellent, thank you for that description of the veterinary aspects of it. The political and business context of this also affects the process, because agriculture has ridiculously narrow profit margins. Chickens in particular, if you're raising chickens for eggs, you are just barely getting by, even if you've got an enormous operation, maybe especially if you've got an enormous operation. So the cost of vaccinating all those chickens is significant. You'd have to go through thousands of chickens and vaccinate them all. You'd have to pay for the vaccine and somebody skilled enough to administer it. On the other hand, basically every country in the world, certainly every developed country in the world, massively subsidizes their farmers. And the U.S. is king of this. I mean, we throw billions of dollars at our farmers. And I'm not objecting to that. I would object to some aspects of it, but this is an ongoing program. And, as this letter points out, the farmers are compensated for the chickens they have to cull, and if they have to cull pigs and if they have to cull cattle, the government's going to pay them for those. So on the one hand, you could pay a lot of your own money out of your non-existent profit margin to vaccinate them, or on the other hand, you could take your chances, and if the virus comes, you just cull them all and you get reimbursed, and it's as good as a sale.
So Angela, you have in Canada H5N1 in chickens, and they have to be culled or whatever kind of bird, yes?
Yes. You'd have to get them culled, yes. The thing is that implementing...
But you don't have the cow H5N1, I understand, right?
As of right now? I don't think so.
Yeah.
But if we did, the cows would have to also get culled.
Well, the U.S., we don't cull the cows because, I don't know, they're not sick. But
then it's still transmitting to the birds and possibly into other animals.
Oh my gosh, cows. I mean, no, they don't cull the cows here. They just wait till they stop making virus and the milk is clean again.
And they pasteurize the milk.
Well, I think from a producing cow, they wait until the infection is over. But why? So here in the U.S., egg prices are going up, right? Why not chicken prices? Why just eggs? Does anyone know?
Broiler farms and egg farms are separate.
I see.
They're not the same supply chain. And there are other factors in the egg supply demand. It's a little less so now that we have more of our flu vaccines being produced in cell culture, but flu vaccine production, hundreds of millions of doses of it, uses a lot of eggs.
It's true.
Traditionally. Easter's coming up, believe it or not. Egg consumption goes up around Easter. And then throw into it the fact that in the U.S. we have this highly consolidated industry that's producing these at massive factory scale farms. My understanding is that in Canada, it's a much more diversified industry. There are more small suppliers feeding into it. And so they're not as susceptible to these shocks. If one farm gets hit, they cull their chickens, but the farm down the road is still tooling along. In the U.S., if one farm gets hit, it's, a gazillion chickens, and that's a big chunk of your egg supply gone. So it's differences in the supply chain in the two countries that have led to the U.S. having this rather significant egg shortage.
Overall, there's just been more chicken farms affected in the U.S. with higher density of chickens, just more actual chickens. And implementing a vaccine in poultry takes a lot of work. Apart from money, actually, like Alan was saying, like physically vaccinating the chickens, because most of the vaccines in chickens are in embryo, like the Newcastle vaccine and these things. Somebody's not like walking around actually vaccinating the chicken most of the time. It's all like in the actual eggs. Having to do that would take a lot of manpower. And then also, what if you don't vaccinate them properly, partial vaccination, then there's like escape variants. There's so many things that could happen that I would not necessarily, I wouldn't want the influenza, like H5N1 being vaccinated against, even though it's unfortunate for the chickens' lives to, for them to die and not to be consumed, because at least they could be consumed. I think it's for the best to control.
But nevertheless, they vaccinate fish. No problem, right?
But vaccinate fish against what? What do you?
Fish viral infections. They routinely take the little.
But those fish viral infections, they're not necessarily going to spill over into us or cause anything in humans or other domesticated species that could then have repercussions in our food chain, etc.
No, that's just to protect the fish so that they can go to market. They don't get infected. Yeah. But it is doable. I mean, they have people pulling the fish out of the bucket.
Oh, for sure. Sure. If it's worth it. But in this case, it's just too dangerous to try to vaccinate. I think.
I understand why we have to cull, but this is crazy, right?
Yeah.
You'd think we'd have a better way or something else.
I know.
Than wasting all that protein.
Yep.
My gosh.
It's true. And those lives, because they're all animals. I mean, in the end, of course, we consume the animals, and I'm not vegetarian, but I still do empathize with the animals that at least we would be consuming them, having given their lives for us, literally. And in this case, they're just going in the garbage and getting incinerated. So it's terrible.
There have been some big cullings of egg-laying farms in the U.S. lately, like 2 million birds. And they said the problem is if you order chicks now to repopulate, you're not going to get them for a year because it takes that long.
Backlog, yeah.
I'm glad I'm not a chicken farmer.
Yeah, I mean, the corollary to the egg shortage is you get a chicken shortage.
Mm-hmm.
You mean there's a chicken egg thing?
Yes, there's a chicken and egg problem here.
Okay. Well, let's see what happens. One more, Alan.
Sure. Anna writes, "Hi all, I've been a long-time listener to TWiV and a big fan of the work you do in science communication. Like many, I am very concerned with the direction science funding is taking in the USA and the global repercussions of obviously short-sighted decisions. I have been part of one of the laboratories in the WHO Global Measles and Rubella Laboratory Network for 12 years now. It has been an honor to work with so many dedicated people that dedicate their time and expertise to help making the world a safer place. The recent withdrawal of the USA from the WHO will have a profound impact in the measles and rubella elimination program, as it will affect vaccination and diagnosis in the poorest countries. WHO is not perfect. It is made of humans, after all. It is the best we have, though. I came across this fundraiser for WHO and thought I would share it with you in case you would like to give it the TWiV bump. I cannot think of a better informed audience for it." And links to this donor support page for WHO. With my best wishes for every one of you, Anna. Thank you, Anna.
$1, One World, WHO Foundation.
Cool.
Yeah, WHO is not perfect, but it's what we have. And,
yes, indeed.
People need to, and you don't quit. You try and fix it, right?
if you quit, you can't do anything about it.
Yep.
All right, let's do some picks of the week. Uplifting picks of the week. Angela, what do you have for us?
So mine is like really cool, horrifying, but cool. So uplifting for science to better understand how cancer, certain cancerous cells are doing their thing, but once again, also kind of horrifying. So this is this paper in Nature. Let me just see the authors. I have to click on so there's a Nature paper, which is also has like a news article. So the last author is Togashi. We'll add it to the show notes so you guys can see. And basically titled Immune Evasion Through Mitochondrial Transfer in the Tumor Microenvironment. So this paper showed that it's the first time anyone's ever showed bidirectional exchange of mitochondria between two cell types. And in this case, it was tumor-infiltrating lymphocytes, so-called TILs, and cancer cells. It's crazy. So if you look at the news article, which we'll also attach, they have this very nice figure where you can see a cancer cell and a T cell. And the cancer cell has, via these tunneling nanotubules between the cells, connects it as if like a little tube is being connected to the T cell and the cancer cell. And the cancer cell siphons off the mitochondria from the T cell and then uses its mitochondria and then gives the T cell this cancer mitochondria, so mutated, let's say, mitochondria that's also marked with USP30 so that it will prevent degradation inside of the T cell, thus causing the T cell to become senescent and ineffective in killing tumor cells. So that's like a lay summary. This is crazy. So I know these things happen, but when you read them like that, literally the tumor cell makes a tunnel between itself and a T cell, gives it really bad mitochondria that it can't degrade, that makes the T cell not be able to kill the cancer cells. And this happens in this study that they did in 15% of the T cells. 15% of the T cells is a lot, especially in the tumor microenvironment. That means that 15% of them can now not kill the cancer cells.
Yeah.
So I thought that this paper and like this news article was just really interesting because of this bidirectional exchange of mitochondria and the implications. So
It's just scary. Like
it's
Yeah.
horrifying.
And this is, these are cells in the body that must have the tools to do this for some evolutionary reason that is not this.
Unclear. Exactly.
Right? So this is a whole other dimension of cell-cell interaction that we didn't know existed.
Like they call them microtubule. What is it? Tunneling nanotube. Horrifying. So I'm guessing it's like part of the cytoplasm and like the microtubules. There must be like some part of the cytoskeleton that's in there. But
yeah.
Wild. I couldn't not share that.
Yeah.
It's really cool. Thank you. Alan, what do you have for us?
Little self-promotion here. So people who visit my alandove.com site, there's a new little tab at the top that just says adventure. And so you can, if you're looking at my site, you can go on a little adventure. I built another game. This is another narrative game. It's just text. Should open on a phone browser just fine. And it's a little adventure you can go on, probably take you about 10 minutes. It has a couple of different endings, one of which could be considered a sort of game over, and the other could be considered a sort of victory. So
It's like D&D, isn't it?
well, to choose your own adventure. So you get a little bit of text, and then you can make a decision, and you can choose what you do along the way and kind of where you go based on clicking the links. And then a story will emerge, and you'll figure out what your mission is and who you are.
I've already started. Wait, Alan, question. Can you see how many people do it and what missions they take? Not like who the person is, obviously, but
Right, so I don't have it set up to collect stats on what endings people get.
That'd be cool.
But I could, if I ever cared to. I usually don't bother to do this. I could look at my website statistics and see how many people have played it.
So you wrote this thing, right?
I wrote this, yes.
Yeah, it's like the old style text games.
Exactly.
Right?
Yeah, and a few years ago, I pointed to another game very similar to this that I made for a contest, a game jam. It's called I Am Promise, where the object of the game is to turn off a robot. And it's just a short narrative experience that had one picture and 1,000 words of text. Those were the parameters for the game jam. This one wasn't for any event like that. It was just a kind of a story I had kicking around in my head, and I didn't know where else to put it, so I turned it into this.
I just keep
That's
picking
fun.
stare vacantly ahead and nothing happens.
Nothing happens.
Stare vacantly ahead.
Ignore
the button. Wait.
Nah, I'm pushing the button.
I like to
Wait.
do all these non-things. I want to see what
yeah,
happens.
but then something's going to happen.
Really?
Yes.
I'm gonna go in multiple times and play multiple times and see what the different outcomes are.
Yeah, there are only two endings. I will tell you
Okay.
that. There is something you can do along the way that will that'll lead to the bad ending, and it'll be pretty obvious with it. It's probably gonna get you there.
Yeah. Yeah, cool. That's neat. Thank you, Alan.
You're welcome.
All right. So this is my pick is a Nature article, The Staggering Success of Vaccines by Tara Halley. All right. So it's got lots of graphics if you'd like to show people what vaccines do, right? Numbers, all different infections, not just viruses. But there's a nice graphic here. Deaths averted with measles, tetanus, pertussis, tuberculosis, hemophilus, etc. Polio. 1.5 million deaths averted. Vaccination rates over time, and just a really good data-rich article on why vaccines are awesome.
Yeah.
And you can show them to people, and they will say, this is lie. These are all lies, they'll say. Because that's all they can say. Disgusting. So if you need to, it's open access. If you need to show people what vaccines actually do, there you go.
93 million deaths averted between 1974 and 2024 just for measles.
Yeah, that's
Amazing,
crazy.
right?
That's a lot of people.
Yeah.
And as
work.
somebody said once at a vaccine conference, if all these people knew who they were, there would be no anti-vaccine movement.
True.
Because you wouldn't be able to say, vaccines are bad, without somebody turning around and saying, I wouldn't be here if it wasn't for vaccines.
Yeah, but you don't know, right?
You don't know. That's the problem. It's an invisible success. It's the tragedy of public health.
Your grandparents, your parents, they great-grandparents, especially as time goes on.
After the RFK hearing, I just realized the reason people are anti-vaxxers is they can make money.
Mm-hmm.
Well, yeah. The ones who are diehards are in it for the Benjamins.
Elizabeth Warren asked him, okay, you are doing all these lawsuits against vaccines and you make money. Are you willing not to take that money? He said, oh, no, I'm taking that money. And then the next day he decided, I'll give it to my son. Like it's any different, you schmuck. But this is the driver, money and nothing else.
Of course.
But it's a quality, it's a flaw because you're sacrificing people's lives to enrich you. You're not even thinking about their lives. All you're thinking about is your pocketbook, and that's disgusting.
Yep.
Mm-hmm. But then when the time comes, just like with Trump during the pandemic, when he was so anti-vaccine, the moment he got COVID, he was the first one to take Paxlovid. Okay.
Yes.
Why? If they're like, it's ridiculous.
But Angela, what is the purpose of censoring scientific information now? Like not putting these H5N1
to keep people
articles.
ignorant. Well, to keep people ignorant and like for them not to know. That's the only reason why I can think they don't want people to know, to have too much information, because then they'll be educated and then they'll make an educated decision, which they don't want them to, because they don't want to listen to the scientists. They don't want us to tell them the truth.
for the past 20 years at least, the Republican Party has been at odds with facts. And that puts them at odds with science. And as a result, here's the culmination of it. They're not reality based.
It's remarkable.
Yeah.
This guy, RFK, he lies. It's clear he's wrong. And yet, I vote for you.
Right.
Mm-hmm.
Holy cow. It's mind-boggling.
It's ridiculous.
All right, we have 2 listener picks. We have John. I have a pick for Black History Month. Since I don't consider myself a virologist, no prominent Black virologists come to mind, but if you expand the net a bit And if you're not already aware of him, I strongly urge you to have a look at steroid chemist, biochemist Percy Lavon Julian. Much is written about him and how he rose against adversity, including bomb threats, to become the first black chemist inducted into the National Academy of Sciences and became economically successful to boot. Hurdles he jumped in getting to that point included needing to sit on the front porch of his Oak Park, Illinois home with a shotgun. His Wikipedia page does a nice job of hitting the highlights. Yesterday,
Oh, sorry.
yeah, go ahead.
Sorry, I thought you were finished. I wasn't looking at the email.
Yesterday, the high in Greater Braddock was 61, and so I managed to do a bit of outside house painting. 61? This afternoon's high is a surprisingly sunny and pleasant 37. The oscillations of spring are starting. Yes, go ahead, Angela.
So I have to add that Black scientist, or virologist, virologist, is one of the developers of the mRNA vaccine, Kizzmekia Corbett. In case anyone forgot,
there we go.
She's like one of the most important ones for sure in the last few years that most people probably know about or have heard us speak about. So yes, there you go. This is the first one I thought of. Amazing.
And Debbie writes, hello to my most hopeful people ever. On Netflix, there's a sci-fi movie called Don't Look Up. It's from 2021 with Leonardo DiCaprio. It's 2.5 hours. Loved it. A must-watch. Kind of covers what's going on today. Same as 2021 Deniers. Hope you'll all watch it. Sincerely, Debbie. P.S. Keep up the awesome work. It's appreciated.
I think
Thank
this
you,
was...
Debbie.
Didn't somebody pick this?
Sounds like it.
I think like Brienne picked it or something. But sure,
I don't remember.
Looked up. Thank you, Debbie. Thank you, John. Thanks, everybody. That is TWiV 1193. You can find show notes at microbe.tv/TWiV. Please send us your questions and comments and pics. TWiV at microbe.tv. We like sharing your pics. It's a lot of fun. Like that snow thing started a little chain, right? People got into it. I'm amazed that Fernando noticed my thumbnail. I thought nobody noticed the thumbnail.
Oh, I noticed the thumbnail.
I actually thought about why I would do that, but I just love that kind of art.
Mm-hmm.
TWiV at microbe.tv. And please, if you enjoy our work, we'd love your financial support. Microbe.tv/contribute. Angela Mingarelli, McGill University Immune Vet on Twitter. Thank you, Angela.
Thank you. I am also on Blue Sky now, also as Immune Vet, in case anybody wants to follow me there.
Should I just put Blue Sky, or you want to put both?
I'll do both. I mean, I'm going to use both. I'm continue to use both. So for
Alan
now.
Dove is at alandove.com, turbidplaque.com. Thank you, Alan.
Thank you. It's always a pleasure.
I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene Ramsey for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.